Good morning, everyone. I'm Simran Da Costa, and on behalf of Alton Technologies, I'd like to welcome you all to our virtual webinar series on pharmacoinformatics. Our session for today is entitled as Drug Design, Synthesis, and Drug Metabolism Prediction using molecular modeling and machine learning methods. So these days, we've been hearing a lot of talks about how computational tools are being used widely for life science related fields to leverage most processes. So machine learning has emerged as a transformative tool in the field of drug metabolism prediction and offers advanced methodologies to predict how drugs are processed in the body. We've seen that traditionally, drug metabolism studies have heavily relied on experimental techniques, which can be time consuming and costly. But thanks to machine learning algorithms, we are moving towards a stage where we can analyze vast amounts of data from various sources to predict how a drug will be metabolized. So let's understand more about this topic from our speaker of the day. It gives me immense pleasure to introduce Dr. Vaibhav Anil Dixit. Dr. Dixit is an assistant professor at National Institute of Pharmaceutical Education and Research, Guwahati, from October 2021 onwards. He has done PhD in medicinal chemistry from NIPER SAS Nagar in 2012. He has completed MPharm in medicinal chemistry in 2006 from NIPER. In 2013, he completed postdoc from Indian Institute of Science, Bangalore, where he performed P450 modeling research work and gave computational modeling support to other projects in the lab. In 2015, he completed postdoc from AstraZeneca, where he worked on the design and synthesis of novel isobutyryl heterocyclanes. From 2015 to 16, he was a senior chemist at Polychem Laboratories, where he was in charge of the instrument section. He did development and validation of analytical methods using LCMS, GCMS, HPLC, and spectrophotometric methods. From 2016 to 2018, he was an associate professor at SVKM's NMIMS Shirpur campus, where he set up computational and synthesis research facilities, project submission to DST, CSIR, and he also conducted teaching. In 2018, he was an associate professor at SVKM's NMIMS Mumbai. There, he developed a computational drug design and synthesis research facility at the School of Pharmacy. He worked on drug metabolism studies using quantum chemistry and PK/PD modeling. In 2018, he was an assistant professor at BITS Pilani. In this role, he utilized machine learning and chemoinformatic tools to develop novel drug toxicity and efficacy prediction models. At present, he is doing multiple teaching, research, and administrative tasks, and also guiding PhD scholars and master students. His research is mainly focused on areas like AI guided anti cancer drug design and drug metabolism prediction, structure ligand based drug design, matched molecular pair analysis for drug design, application of quantum chemistry, molecular docking, and dynamics to drug design, application of organic chemistry and electrochemistry for drug synthesis. So, Dr. Dixit's lab focuses on anti-cancer drug design and synthesis, specifically of PARP1 inhibitors, drug metabolism prediction, and heme protein re-engineering for biotechnological applications. In his lab, they perform drug design, binding free energy, and quantum chemical calculations to solve problems in various fields, including organic reaction mechanisms, drug metabolism, and toxicity predictions. They also employ quantum chemical and machine learning methods to assess the potential mechanism for impurity generation in drug formulations and during storage. His group also develops in-house drug discovery workflows and softwares using both open source and commercial tools and performs modern computational chemistry drug design and machine learning tasks. Additionally, his group has also developed novel green chemistry matrix and a web tool for assessing drug synthesis route greenness. They also use electrochemistry for synthesis. So, without taking much time, I'd like to hand over the platform to our expert. So, welcome, sir, and I invite you to take over the session.
Thank you very much for that elaborate introduction. And without taking further time, I would like to share my screen and start the presentation. So, I believe you are able to see the screen. Hello.
Yes, sir. Yes, sir. We can hear you.
Yeah. Are you able to see the screen as well?
Yes, sir. Yes, sir. We are
Hello.
able to see your screen.
Yeah, great. So at the first outset, let me thank the Altem technology team. We have been working with them for the last couple of years or more, and they have been also supporting us in our work. So thank you very much for giving me this chance to showcase our work and also talk about the topic pharmacoinformatics. So today my topic, as she mentioned, will be on drug design, synthesis, and metabolism prediction using modeling and machine learning methods. So, to begin with, I would like to give some background to the student audience about how computational sciences is to be seen. And we can see that there are different biological systems that can be studied at variety of levels. We people do systems biology, standard biology studies, computational biology. Then there is bioinformatics and informatics. So all these fields overlap extensively, and it leads to sometimes confusion about what is bioinformatics, what is informatics, what is computational biology. So this is one of the way you can view different fields and how they overlap with each other. So let me begin by trying to define what is pharmacoinformatics. This term was coined by Professor Bhartam, who is a professor and head of the department at Nagar, SAS Nagar. He coined this term in 2003 and started the Department of Pharmacoinformatics, which has now spread its arms and widths all across the country and outside also. Many students uh, and professionals who are very successful in this field, they are working across the globe in various positions. So to begin with, pharmaco means something related to pharmaceutical sciences, and informatics means the study of structure, behavior, and interactions of these natural or engineered systems through computational means. So when you combine these two, you get pharmacoinformatics, and it can be defined as a discipline where the structure, behavior, and interactions of pharmaceutically important systems or processes are studying using various modeling methods. And those studies can be done at different levels. Those levels could be at atomic, molecular, cellular, tissue level, or organism, or it could be at a higher level where you are studying changes in the population or the whole environment. So this, what does this modeling means? It means a simplified or idealized description of a system that allows you to calculate properties and make predictions. So again, as here is the definition as given by Oxford English Dictionary. And so whether you know or not, you must have made models, whether you are studying biology or in other areas of pharmaceutical sciences or chemistry, you have developed models. And models like these uh, and these, so we all think them, which one of is the real Taj Mahal? None of these is the real Taj Mahal. All of them are models. And so whenever you have drawn a cell or you have drawn a structure like this one or this one or a 3D model, all these are models, and they try to represent the pure compound because humans can't see these molecules, they are very tiny. So we end up developing models so to develop an understanding of these systems and how to expect what behavior they will show. So what is it that we want to model? So depending on the field in which you are working, if you are working in biological sciences, you will be interested in modeling metabolic pathways, cellular processes. If you are working in chemistry, mostly we are interested in the structure and properties and how reactions are happening between molecules, how do drugs interact with receptors, what are their rates of these reactions, and what are the different mechanisms. In the area of physics, people are mostly interested in the study of matters and systems. In mathematics and statistics, people study relationship between different variables. So, for example, one can easily identify what will be the average height of a child or infant as they grow older, and you may plot that data in the Excel sheet, and you will get this model uh, simple in the Excel sheet. So, if I know the age of the person, I can know their height. So, can I apply this model to my particular case? Suppose I have a child and I want to predict what will be their height at the age of 30. Can I simply use this model to predict their height? If I do so, I end up getting a very absurd answer. So, meaning to say that all models have applicability domain. They should be applied within the applicability domain. And if you really want to know what will be the height of a person when they are small infant, then that is very difficult. But you could certainly predict their height at puberty, what will be their height as an adult. So this type of modeling is done by everybody. Models are everywhere around us, whether we realize or not. So with this, we can go back to chemistry. In chemistry, we are mostly interested in energy. We try to predict energy by using laws of physics like Newton's laws, its equations, Leonard-Jones potential, Faraday's laws of electricity, electrodynamics. We apply these laws on atoms and molecules so that we can predict their energy. So, what type of energy we are mostly interested in chemistry is the potential energy, because energy is two types, kinetic and potential. But in chemistry, mostly we are interested in potential energy. Broadly, there are two ways of calculating potential energy: molecular mechanics and quantum mechanics. And molecular mechanics is widely used in organic chemistry. For example, we could find a relation, we could develop a model to find the relationship between bond distance and energy. For the simplest of the molecules, hydrogen molecule, you will find that the relation is something like this. And it can be summarized into an equation which has a bunch of parameters and values that go into it. Similarly, you could write a model for predicting how the energy of the molecule will change as the bond angle changes. So for that, this type of a function from Hooke's law is possible. For even for torsional angles, we could write a formulation with a cosine function like this, and all those terms put together gives you something known as force field. It has the bonding terms and angle terms and the non-bonded terms. This is the Lennard-Jones potential, and this is the Coulomb's term. So these force fields, they can be thought of as something that you see in movies often as a field around you. So these fields are very similar conceptually to those fields. So moving to some examples, and in the area of quantum mechanics, we also predict energy, but this time we account for all the electrons and nuclei that are present in these organic molecules. And then we could, once we know the energy, we could use it to optimize the energy and do many other tasks, for which I will show you examples. Here are some of the popularly used softwares. Some of them are open source, some are commercial. You could use them to do quantum chemical calculations. And you could also use other visualizing tools like WebMO, Avogadro, and uh, our host Discovery Studio and Altem people. They also provide a lot of visualization tools for getting these tasks done. So at Niperg, we use these tools for teaching our students and also doing our research work. Behind these quantum chemical calculation, there is a Schrodinger equation. In the interest of time, I am not going into the details of how this equation is solved by these softwares to get the energy. So here is an example how these molecular mechanics and quantum mechanics based tools can be used for tasks like geometry optimization and energy minimization. For example, if there is a molecule which contains n atoms, its degrees of freedom is decided by the internal coordinates. So for a molecule like ethane, it already has 18 internal degrees of freedom around which you need to optimize the geometry and get the potential energy surface, which may look something like a hill which has valleys and troughs. And So these are the energy as a function of different coordinates in the molecule. So for ethane, it is not very steep, but very similar energy. Many conformations will have very similar energies. So for example, you might immediately recognize this molecule as methane, looking almost like a methane, but something is wrong with it. That is the carbon hydrogen bond distance. So once we apply these optimization tools and methods like the first derivative, steepest descent or conjugate gradient, then there are other second derivative methods and more useful information about how to think about optimization you can find on YouTube. So once we apply those optimization algorithms, we get the correct structure. So similar type of calculations you could do even on higher, bigger molecules, and you might see how the molecule is rearranging itself as it is trying to find the optimum geometry that is of lower energy. So, so these optimization and energy minimization methods we have applied. You could easily apply them for doing frequency analysis, IR frequencies, Raman frequencies. You can calculate them. You can also determine the transition state barriers for path reactions like SN2 reactions. Simple, these we train our students every semester for these. Earlier, we have studied the metabolism of troglycazone and tried to explain that which of these functional groups in the molecules they lead to toxicity. And we identified some unique metabolic pathways that lead to the formation of quinone methydes and explain the toxicity of this molecule. And this is the transition, one of the transition state that leads to the formation of the quinone methide reactive metabolite in this drug. And this work is published long time ago. You interested people can go in the details. But now I am switching gear and I am going to show you some recent applications of molecular modeling and drug discovery that we are doing currently at NIPER Guwahati. So mostly, we, like many other people, we are interested in breast cancer drug discovery. This is the project which has been funded by MIT and DOP jointly. And as you know, that breast cancer is affecting a lot of people around the globe, and there are multiple treatment options. Particularly, we are interested in the PARP inhibitors, for which 4 drugs are available in the market currently. But they have some limitations that they are very costly and there are selectivity issues. And recently, they have been shown to be cardio and hematotoxic. So we have a project in which we are identifying hits to avoid these toxicities, and we are also doing synthesis, and in vitro activity is underway as I speak. But I will walk you through the work of some of the PhD scholars who are working with me, and we have done literature search and understood that uh, there are multiple drug-like properties that are important for the successful drug discovery programs. So there should be optimum balance between these properties, physicochemical, pharmacokinetic, and dynamic. And there obviously there are drug likeness rules are available, but these set very hard cutoffs that often lead to rejection of potentially useful compounds. Therefore, multi-parameter optimization should be done with soft cutoffs known as desirability functions. And we analyzed the known literature on these PARP inhibitors, and we developed our own multi-parameter optimization desirability functions. So I won't go into the details in the interest of time, but you could read this article and find out more how we have defined these utility functions that allow us to separate the toxic and non-toxic molecules, where we came up with the D score, and it is a multiplication of the different utility scores for PARP1 inhibition, cytochrome P450 inhibition, hERG inhibition, and the 10 kinase inhibitions. Later on, we collected more data from the literature and applied some standard guidelines for data collection and curation. Collected data only for single protein with high confidence level. That gave us around 4,300 compounds, for which we calculated descriptors using ELVA test. And then we removed low variance and highly correlated descriptors. Then further, we found that the data was not balanced because most of the compounds were having high potency and lower potency compounds were not available. So we used a method that is popularly used in machine learning areas known as synthetic minority oversampling, that creates artificial balance between actives and inactives. Then the data set is according to standard OECD guidelines. We did the splitting of the data set, applied 10 machine learning algorithms, and developed models which were further validated with standard machine learning metrics. So here is a summary of those results. And we found that out of the 10, KNN is the best performing algorithm. With the use of 15 key descriptors, it is giving us best performance of the balance of sensitivity and specificity. Therefore, we have taken forward this model and identified compounds which are known to be, which are likely to be active. Then we have also developed regression models using two data sets which had some around 700 compounds and 70 compounds each. And so these are more focused. They have narrow applicability domain, these regression models, but nonetheless they are useful. And we found that the support vector machine and multiple linear regression is allowing us to get high accuracies of prediction and lower errors. So these were later used in other projects to make predictions. But additionally, we also are interested in another very key medicinal chemistry method known as matched molecular pair analysis. It will address how the question of how to improve PARP1 inhibition, because the typical QSAR methods, they do explain the activity trends, but they do not directly answer this one important question, that what structural modifications a medicinal chemist should do to further improve the activity. Therefore, we have identified matched molecular pairs, which are well defined pairs of compounds which differ only by a small change in the structure, as you can see in this example. Using these matched molecular pairs, the analysis is performed for systematic generation of trends and changes in property identification. Once we do that analysis is known as matched molecular pair analysis. It has been widely used in pharma industry for lead optimization and multi-parameter optimization and even toxicity predictions. So when we do this at a global level on all the data set, it does predict property change, but global MMPA has the limitation that it attributes all the activity changes to the substructural transformation that is happening within the molecule. So it is, in other words, it is telling that the activity changes are not driven by the rest of the molecule, but only by this small change in structure, which already looks like an oversimplification. Therefore, it often ends up giving incorrect interpretation. Therefore, we have recently in one of our studies, we have done context-based matched molecular pair analysis on the PARP1 inhibitor dataset. NIME Analytics platform was used extensively for developing this workflow. I won't go into the details of this workflow in the interest of time, but to summarize the results, we found that it is important to separate the actives and not active compounds based on their scaffolds which they represent. So, for example, phthalazinones and quinazolines, they experience an increase in activity if you substitute hydrogen with fluorine. But the tricyclic class of compounds, they experience the opposite effect. They show a decrease in activity if we substitute hydrogen with fluorine. So these type of predictions are not possible with classical global MMPA, but these kind of key medicinal chemistry insights can be derived from context-based MMPA, and we have recently published this work in the Molecular Diversity Journal. And all this methodology of classifying the unknown compound and trying to predict its activity has been deployed as a web server, which is available on this website, and you can access that and try to check the activity of your compounds if you are interested in PARP1 inhibition activities. Recently, this web server was launched with at NIPER Hyderabad conference by the hands of Honorable Secretary DOP and our own director, Professor Murthy. So, in addition to this, we have been doing other type of virtual screening for PARP1 activity predictions, and those we have combined with our ML models that I just discussed. Using those, we have identified these triazolopyridine class of compounds. They show a lot of promise for PARP1 inhibition. Thus, we have used synthesis schemes we have designed, and we are applying a combination of Suzuki coupling and other types of copper catalyzed reaction to get to the desired compound in 2 steps. And the synthesis of some compounds is already done. and we are actively in the process of testing the activity of these compounds in the lab. But as we know that synthesis of these compounds is very challenging, it leaves a huge negative environmental impacts because you end up generating a lot of waste during the synthesis. And but there are green chemistry principle concepts are already available that allow us to reduce the impacts. But most of the time, the key challenge is the unavailability of the data, and therefore we have tried our own method to tackle this problem and came up with cumulative green chemistry principle score that is based on the four key green chemistry principles of atom economy, less hazardous synthesis, and green solvents. So in this paper, we have detailed the methodology of these that is behind the ACGCP and CGCP scores. And once you analyze the retrosynthesis and synthesis scheme in detail, then you can assign the scores to the different steps in the scheme. And we have validated our CGCP score using a set of 11, 12 drugs which are known to be green. Their synthesis routes are known to be green. Then the same methodology was applied to PARP inhibitors, and as you can compare, the most green route for sildenafil gets a score of around 0.6, but all the PARP inhibitors are having a lower score. That means there is a scope to further improve their synthesis procedure and make it more green. This analysis is only possible through our CGCP score, and to ease the calculation of this score, we have also developed a web tool that is accessible through these links. So now I am shifting gears and moving to the drug metabolism prediction area. In this topic also, we are actively working and applying machine learning tools to solve the drug metabolism prediction problem, because it has strong connections with toxicity. As we know, that drug metabolism mostly happens for the elimination of foreign compounds, but in the process, it might end up making molecules more polar. And there are two types of drug metabolism processes classified into phase 1 and phase 2. Phase 1 mostly involves oxidation, phase 2 involves conjugation. So this is well known facts that we study in the somewhere in the B from third year or similar. But here is the overall fate that we recently summarized in one of our review articles. Drugs, they usually end up being non-polar, and they usually interact with their targets. They give expected therapeutic benefit. But whenever they interact with non-desired targets, or known as off targets, they give secondary pharmacology and side effect. Drugs also undergo metabolism simultaneously. Mostly this metabolism is phase 1 and phase 2. If the metabolites are polar, they are easily excreted out of the body and there is no side effect. But every now and then these metabolites undergo another round of metabolism. As long as these metabolites are still lower polarity, they may undergo conversion into reactive metabolites that will precipitate toxicity. So when we understood all this, we decided that not only cytochromes but other phase 2 enzyme prediction methodologies are also required. So since then we are working in the area of drug metabolism prediction, and as you know, site of metabolism is an important concept in this area, which can be defined as an atom or a group of atoms where metabolic reactions are initiated. So if you can see, very simple example, phenacetin undergoes metabolism at the carbon, gets converted into paracetamol, which undergoes metabolism at the oxygen, get converted into reactive metabolite. So by looking at this, you will say that, oh, predicting metabolism is relatively easy. So one could do that. But no, guessing site of metabolism is non-trivial. For example, in the case of dopamine, can you predict which of these groups will undergo metabolism, whether this hydroxyl, this, or some of the benzene carbons, or the nitrogen in the molecule? So the site of metabolism is any one of those hydroxyl groups. And even the in vitro liver microsomal incubation followed by LCMSMS methodology does not always allow you to unequivocally tell where the metabolism takes place. Therefore, these site of metabolism annotations are, and the important kinetic parameters, these are very important in the study of drug metabolism reactions and drug metabolism predictions. So this is the overview of what is the current state, how many of the drugs are metabolized by phase 1, phase 2 enzymes, and their relative contribution So the major focus in any drug discovery project is on the phase 1 enzymes and cytochrome P450s. But needless to say that the phase 2 enzymes cannot be ignored. They also should be studied. So because cytochromes are the most important ones, we first talk about that. And in this cytochrome P450 catalytic cycle is really ends up being somewhat complicated. These are monooxygenase enzyme. They transfer one oxygen atom to the substrate. In this process, they require reducing equivalents from the reductase enzymes, and they catalyze CH, N, and S oxidations. Overall, the reaction ends up being a addition of the OH group on the RH compound. So here is the complicated catalytic cycle. Without going into the details, I would like to summarize that the first step of electron transfer is rate determining. And here is a overall picture of the reductase bound to the bacterial P450. So this electron transfer takes from the amphetamine to the heme after the ligand is bound to the active site. So it is the rate determining step is the most important step in the P450 catalytic cycle. If one can predict that accurately, then you might be able to predict whether the drug will undergo metabolism or not. So therefore, we have been interested in these electron transport processes and rates, and we have tried to understand how different types of ligands, whether they are small molecules or even metal atoms, how do they affect the cytochrome P450 inhibition. So we have used the very famous Marcus electron transfer theory, which allows you to calculate the reorganization energies and the free energies of the metabolic reactions and electron transfer steps. And we are successfully able to explain the differences in the behavior of monovalent cations and the divalent cations, and even the type 2 inhibitors their behavior can be explained using this Marcus treatment for electron transfer theory. Those who interested in the details can read these papers and can also write to me directly. I will be more than happy to explain your queries on any of these topics that I have discussed. So additionally, we have been interested in other heme proteins because they are also electron transfer proteins. They are very important in the synthesis of high value material, and because uh, the retrosynthetic classical methods, they end up being very costly, low yielding, and non-selective, and non-green. So there is always interest in the study of alternative methods, and how, but there is a lack of mechanistic understanding that avoids this full exploitation of the bacterial P450s. The structure based methods and directed evolution, they are useful, but they have their own limits, and Marcus theory was never applied earlier to this problem. Therefore we decided to apply Marcus theory for the estimation of electron transport rate. We studied initially through molecular docking. Can we explain the behavior of these well-known substrates on the bacterial cytochromes? And there is some good correlation that one can see. And MD simulation coupled to the docking is has allowed us to explain the electron transfer rates. These are the experimental electron transfer rates, and these are the calculated ones. They are within 1 log unit, and that is considered as a reasonably acceptable uh, uh, prediction. And we are able to explain the effect of mutation, and also able to explain the effect of ligands on these electron transfer rates. And this work is also published a couple of years ago. So, and this there is also a web tool and GitHub code is available if you are interested in predicting these electron transport rates for cytochrome P450 enzymes. So moving forward to the phase 2 enzymes, which one of the student has recently completed this part of the work, we are interested in sulfotransferase because we noticed that no reliable method is available in the literature to predict this complicated sulfonation reaction. So we focused our attention onto that. We collected the literature data available, and we did painstakingly, we manually did the annotation. We used molecular docking and bond dissociation energy calculations at the semi-empirical level. Using that data, we developed models based on accessibility and reactivity, and we are able to generate good models. So accessibility, reactivity, and hybrid models are reaching true positive rate predictions that are close to 90%, and in some places they are still close to 80%. So these 3 or 4 models were handpicked after thorough analysis, and we developed an algorithm through which you need to just put your molecule as a SDF file. It will first check whether it contains a hydroxyl group. If there are no hydroxyl group, the molecule is rejected. It cannot be sulfonated. If it contains hydroxyl groups, then we apply some rules whether the molecule is steroid or opioid. Then we do semi-empirical calculation, docking calculations, and then we pass it through this algorithm. If the cutoff parameter values are within the cutoff, we declare that those hydroxyl groups are the site of sulfonation. If none of the parameters are within the cutoff, then we have to revert the class of the molecule and declare that this molecule is not a substrate. All this detailed story is available in the recently published paper, and also the SOS predictor is available as a web tool where you can simply upload your SDF file and hit the button of SOS prediction. It will give you this type of classification that within this molecule there are two OH group. Only one of the OH group is sulfonated, and the details of the methodology can be found in this recent paper. So all this complicated work that we have been doing has been generously supported by Ministry of Electronics and Information Technology, mighty funded project. And we have, using that support and support from the DOP, we have developed a supercomputing facility at NIPER Guwahati at the Advanced Center of Computer Aided Drug Design. And we have conducted a multiple conferences over the last 3 years where pioneers in this field came from AstraZeneca, from Triple IT Hyderabad, from NIS Jorat, and from our own NIPER Mohali. Professors came and they have delivered talks. So we are very much grateful to the funders, Royal Society of Chemistry, Ministry of Electronics and Information Technology, DOP, NSM, DST, whose cytochrome work is funded by that. We are also thankful to our director and NIPER administration for continuously supporting the group, which you can see here, the nice people. Those all the work that I am able to show, it is their hard work for the past 4-5 years. And with that, I thank you once again, the LTEM team, who has also been supporting us. Sometimes our license get expired, but they keep supporting us through their generous support, and so that our work doesn't halted till the time we deal with complicated purchase procedures to get the work done. And with that, I thank you everyone, and we are open to any questions and queries that the audience may have. Thank you. Hello. You are muted, I guess.
Yeah, so thank you so much, sir, for such an insightful talk on machine learning, on drug metabolism, and also thank you so much for going into the depth of it. So it's time to take some questions now. If you have any questions, please post them in the chat. Our first question is, can we assess bioactivity of a compound using SMOTE?
SMOT is actually a machine learning method. It will artificially create actives and inactives and balance them if the original data set is not balanced. So short answer is no. On its own, it does not predict the activity, but it helps you build a model that can give you the prediction of the activity.
Okay, sir. Next question, please. Is MMPA different from fingerprints in molecules/ligands used for predicting toxicity in Discovery Studio?
Sorry, can you please repeat that?
Yeah. Is MMPA different from fingerprints in molecules/ligands used for predicting toxicity in Discovery Studio?
Yes, MMPA is quite different because it is like an inverse QSAR method where no descriptors of any kind are calculated. Only differences between two very closely structurally similar compounds, differences in their activity are calculated from the experimental data, and they are averaged over a larger data set. So this is totally different.
Yes.
It's not like calculating descriptors.
Thank you. Can we have the next question, please? How to model metabolic pathways?
Yes, we are also actively working in this area. We have developed our own tools to model metabolic pathways. The work is in progress, but there are already other tools available that who somebody who urgently wants some answer, they can go to websites like Xenocyte or Biotransformer, and some of these tools they can be used. I believe Discovery Studio, if it has any metabolism prediction tools, you are the best person to answer that.
Can ligand protein interactions be understood with mesh molecular pair analysis?
Not directly, but somehow you might use it to do a retrospective analysis, as we have done recently in one of the work that I did not show. It is still under review, so maybe by the end of this month it will be available. And as soon as it is available, I will post it on my LinkedIn page. So you may want to track that. So indirectly, yes, but not directly.
Do we have any more questions? How do molecular docking techniques contribute to drug design?
No point. That is quite a yeah. Uh, So molecular docking alone, it cannot predict activity with confidence. It has to be used along with a data set of known actives and inactives. Then you need to develop your docking methodology, and then you need to do enrichment analysis. That means you need to do validation on a given data set. So molecular docking cannot be used in isolation for predicting activity. It is just like any other modeling method. It needs thorough validation, uh, and meaning that within a given set of actives and inactives, is it able to correctly always rank the actives as top scoring compounds. So once you have a model that is able to actively correctly predict the actives and correctly predict the inactives as inactives, that means it has a good balance of sensitivity and specificity, then it can start contributing to drug design. But without validation, it has very limited value.
Okay. Thank you, sir. Do we have any more questions? Machine learning algorithm and deep learning algorithm are different from each other. Which is the best model for drug design, like drug affinity prediction?
Yes. Again, this question is very similar because there is no one answer that suits all. It will be heavily dependent on the data set and the type of target you are working on. So the more data you have, the better your machine learning model will be able to capture the differences and the similarities between compounds, actives and inactives. So deep learning methods are useful only when you have huge data sets. But standard QSAR and machine learning methods can be used on a smaller data set. For example, like a collection of 50 compounds or 100 compounds. If you have those, then you use classical QSAR methods. If you have a data set of hundreds of compounds, then it makes sense trying to develop modern deep learning methods or models.
Can we have the next? Yeah, thank you, sir. Can we have the next question, please? Okay, I think we are done with our questions, sir.
Yes, I hope I was able
So,
to give satisfactory answer to the audience. Thank you.
yeah. So, thank you so much, sir. So, it's now time to felicitate our speaker of the day with a certificate of appreciation.
Oh, wow. Thank you. Thank you so much.
Yeah, so, thank you so much once again, sir, for giving your time, and thank you for all of your valuable perspectives that you've shared with us and also our audience, and for the great interaction. It was really a pleasure to have you with us here today.
Bye.
Thank you, sir. Also, thank you so much to the participants as well. If you guys want more such sessions, please subscribe to our channel and also please do leave a like. So, thank you so much.